for the last bit of the talk, I'm going to talk about yeast that I'm forced to mate sexually. It's not that hard, but it's just not what a lot of people do. So, when we want to introduce, so the chance of passing on this particular trait, which is we'll call this like the red trait, the chance of passing it on is only about 50%. However, when you have a population that's mostly yellow cells, what happens to the red trait is over time it gets diluted out in the population. So one of the challenges with rare or engineered traits when trying to modify population in sexual populations is that they get diluted out if they're not selected for. And that's where gene drives come into play. So gene drives are selfish genes that bias their own inheritance. And one method that they employ to do this is with endonucleases. And what we wanted to test is that if we could use CRISPR endonucleases, so the system I described before, to make gene drives. So if you take this example here, where you have a gene drive parent and you cross it with a wild type parent, what happens is the progeny should have one copy of the chromosome from the gene drive parent and one copy from the wild type parent. What happens with the gene drive is that an endonuclease in the gene drive is expressed, cleaves the wild type locus, stimulates homologous recombination between these two loci, and the result is a gene-driven prodigy. So the result is a homozygous gene drive progeny. So if we look at that original cartoon with the red yeast and the yellow yeast, if you had the red yeast's allele, or the trait encoded by a gene drive, what would happen is the progeny would be biased to have the gene drive trait, so greater than 50%. And over time, even if it was mating in a population that was mostly wild type, over time what would happen, we'd hopefully get the population being taken over by the gene drive. So these are very powerful and very interesting. And we wanted to test if we could make these using the CRISPR-Cas system in yeast. So basically, they call them gene drives because they can drive gene through populations. That's where the drive comes from. So safety is a very important consideration when working with gene drives because they have this ability to drive through populations. So we took two different approaches for safety. And the first approach was separating the Cas9 protein from the gRNA. And if you remember, the CRISPR system is a two-component system in which you have the gRNA and the Cas9 protein set. As two separate components. So what we did is encode the Cas9 protein on a plasmid and incorporate the gRNA into the locus. And what the gRNA effectively was the gene drive itself. So the reason we put Cas9 on a plasmid was because plasmids can be, if they're not selected for, are eventually lost in yeast when they reproduce either sexually or asexually. So this was important in case of the worst case scenario when some of these strains got into the wild. Over time, they would lose the Cas9 plasmid and in effect lose the ability to drive the gene drive through the population. So we did a really cool proof of concept experiment where we took the AID2 gene and designed a gene drive to knock it out. And we chose the AID2 gene because of this really cool phenotype. So when cells have an AID2 gene, so when they're prototrophic for AID2, they are white. But when they have a knockout of the gene, they are red. And this is because of accumulation of a particular metabolic intermediate. So this was a really cool readout for either having a gene drive, meaning red cells, or not having a gene drive, would mean white cells. Also, when the cells were gene drive cells, these red cells can't grow in media without adenine. So that was another check to make sure that they were correctly mutated by the gene drive. So we tested two different gene drives at the AID2 locus. The first one was just a guide RNA that completely knocked out the wild type AID2 gene. And this would turn the white cells, or yellow here, into red. And so the cells would become red, and they also would not be able to grow without adenine. The second gene drive we designed contained a cargo gene, a UR3 gene. And we wanted to test this system because it if you wanted to spread a gene throughout a population and not just knock out a gene, if you wanted to test that that was actually possible. So this URA3 gene, so these cells also become red, don't grow without adenine, but they can grow without uracil. So these cells, if the gene drive worked correctly, would be able to grow without uracil, so they get a trait with this gene drive instead of just having a knockout. So just as a refresher for different, the two different types of inheritance. With normal inheritance, the chance of getting the red trait would be 50-50, but in the gene drive case, the chance of getting, we would hope that the chance would be much higher to get the red trait. So the ideal would be 100%. So what we did is we did these particular matings with the wild type strain and the gene drive strain. And then I did tetrad dissection, so I looked at the 4 progeny resulting from one mating. And what we found is when we looked at 15 different crosses, that all of the crosses were red, indicating that the gene drives were working quite well. And we did some different controls where you had in which there was no target for the gRNA, so this is where the scissors couldn't cut. And what we found is you get that expected normal inheritance phenotype where you get 50-50 white and red. And then also in a strain, in a cross where we didn't include the Cas9 protein, which is essential for cleavage, you also get a 50-50 white and red. So this was great. That meant that indicated to us that gene drives were working quite well. We also looked at the URA3 cross, and we found that those also had that red phenotype. All of the tetrads I dissected had that red phenotype. We also plated them on media without uracil. So if the gene drive worked correctly, we should see that the progeny have a URA3 gene and they can grow without uracil. And we found that they actually do. They grow very well without uracil. If you compare that to the wild type parent that didn't have URA3, it can't really grow well without uracil. It's hard to tell in these plates, but trust me, they can grow without uracil. We then did a qPCR to look at the population level. So instead of just looking at 15 crosses, we looked at thousands of crosses. And what we found is that the gene drive was biasing inheritance by over 99%. So that was great. That was really exciting. We then wanted to see if we could take these gene drives and cross them into many different wild type strains. So to show just how far these gene drives could cross into different strains. So we took this parent strain and crossed it with 7 different wild type strains of diverse backgrounds. And what we found is a similar over 99% biasing in all the crosses that we did, which is pretty powerful. And it was very impressive that it actually worked so well. So just as a refresher, the approach that we first took was to separate the gRNA from the Cas9. But the issue with this particular approach is that these aren't complete gene drives, so that the gRNA is not actually associated with Cas9. So in a second approach, we associated the Cas9 genetically with the gRNA. However, without any other modification to the locus, this would be very dangerous, because if this was released into the population, into the wild type population, this is completely autonomous and could spread by itself. So what we did is we recoded the locus that it was targeting, so that this complete gene drive could only target specific lab strains. So this was contained in that way. So this particular construct could only target engineered lab strains. So we designed 2 constructs using this architecture. The first one was just this plain construct using Cas9 and a gRNA as a complete drive, or a complete or autonomous drive. And the second construct was a reversal drive. And this is a really cool drive because it can reverse the phenotype of the previous drive. So in this particular, the first complete autonomous drive, it knocked out the A2 locus, as the previous drives did as well. But the reversal drive would actually remove the autonomous drive, and restore the A2 plus phenotype. So these cells would turn white again, and they could also grow without adenine. So this is just a little close-up of what's happening with the complete gene drive. So you have the Cas9 gene and the gRNA, and then this gRNA guides the Cas9 protein to cut at this particular recoded locus. So this is the area in the genome that's not found in wild-type cells. So this is a lab strain. Then this stimulates homologous recombination, and you get a replacement. So using qPCR to look at the population, we found that the gene drive was able to bias inheritance by over 99% as well. So that's great. So the complete gene drives work very well, as well as the separated gene drives. And then when we took the reversal drive, so the reversal drives I think are super cool because they restore the effects of the previous drives, and they would replace the autonomous drive and restore the 82+ phenotype. And these are super cool because if someone was to use gene drives to modify a population, it's important to know that you could reverse the change that you made. So you could use potentially reversal drives to do this. And the way they work, the way that the one we designed worked, is that the guide RNA cuts a region at the end of the complete gene drive, and this whole region of the Cas9 and guide RNA and the wild 82 locus is incorporated into this region, and that's homology to replace the complete gene drive. So what happens is you get expression of the guide RNA stimulating cleavage here, and then recombination is stimulated to completely replace this locus with this locus. So in the end, you have a wild type 82 gene with the reversal drive. So that one worked very well as well. So as expected, I guess, at this point. But and the inheritance was biased by over 98%. So then what we did too is we did the tetride dissections that I showed for the other drives, and we plated them on media without adenine. And we showed that these strains grow very well without adenine. If you compare them to the complete gene drive parent and the reversal drive parent, they all grow just as well as the reversal drive parent does. This one, it does grow. It's hard to tell. This one's a little funky, but there are colonies that are growing quite well in there too. I think it just has a slow phenotype. So that was great. It proved that the reversal drive was working quite well. So a summary of the yeast gene drive experiments I did. We demonstrated that CRISPR-Cas-based gene drives function in yeast. We tested 2 different containment methods. And we also showed that the phenotypes could be reversed. And this was important because no one had tried to do Cas9-based gene drives in any organism before, or in yeast at least, or in any eukaryote. So this was a cool proof of concept for eukaryotes. So as a conclusion, with my PhD, I was able to develop a novel strain that incorporated all of the nucleotides very well. That was Yogi. I demonstrated that CRISPR-Cas systems can be used for genome engineering in yeast. And I also explored the use of CRISPR-Cas systems to bias inheritance in gene drives in yeast. So some future applications of some of the work I've done. Cas9-mediated genome engineering could be really powerful for incorporating multiple genes into genomes without selection of the locus, as I showed before, about 100%. And then Yogi could be potentially used to modify these genes and pathways. So these 2 techniques could be used quite well in concert together. Also, some of the future applications of the gene drives in yeast. It's possible that you could use, we're actually doing it, people in the lab are doing this now, where you're taking multiple gene drives and doing multiple knockouts using 2 different gene drives. And this technique could be expanded even further to make a gene drive for every gene in the genome and potentially make minimal genomes using gene drives and knock out all the genes that are not essential. So if you had 2 genes, a red and a blue gene, over time in a population, you could slowly get takeover by both of the purple phenotype maybe. And what we're doing, actually, I'm doing with a collaborator, Alex Chavez and Rebecca Shapiro, is applying this to an important pathogenic yeast, to Candida albicans, to hopefully expand the double knockout library in Candida albicans, which is a yeast that's kind of hard to engineer right now. So the future of genome engineering is bright. That's how I'm saying. You can do all sorts of stuff. So some of the awesome things that are happening in our lab currently, so you are modifying pigs to be more fully humanized so that you could use pigs as complete organ donors. Use gene drives to make resistant mosquitoes that can't carry malaria anymore. Andy in our lab is working on that. Or you could do something as awesome and fantastical as turn an elephant genome into a mammoth genome and people in our lab are working on that as well. So there's tons of different things you can do with genome engineering. Or at least with the elephant, get a cold-resistant elephant. That's the first goal. So I want to provide some acknowledgments to some people that have helped me along the way. So I first want to thank my committee members, Cleo Wilson, Wong, Jim Collins, and James Galligan for being part of my committee, as well as my two my university that my program is in, BU, as well as the genetics department at Harvard, who all provided such great support and help through the years. And I really want to thank George for providing such a unique environment in the lab. George is really a guiding light that guides all the projects in the lab. And it's impressive how well you can keep track of all these projects and know the literature so well. I'll always be grateful for the time that I had in the lab. And it laid the groundwork for my life as a scientist. This is George, and this is an older picture of George. I found it in a biotechniques paper, that was pretty cool. And then I had the honor of playing George in our skit for his birthday last night. And it was one of the greatest acting experiences of my life. I really want to thank some people that have helped make all this science happen. Harris Wang was one of my first mentors in the lab, and he was my first contact in the lab, and I said I wanted to work on yeast and do something like MAGE, which is this project he had originally worked on, and he said, "That's great, come work with us." And so it was awesome. He really helped form how I make experiments and do them in very efficient ways. So I'll always be grateful to Harris for that. Really want to thank Andrew Conley, who was a visiting scientist in our lab, and he worked a lot with the yogi work that I showed in the first part. He works at VTT in Finland. He visited our lab for, I think, only like 6 months, but he did so much work we did so much work together that it was just amazing. I just want to thank Alex, who's been a mentor for me with so many experiments, especially CRISPR-related ones and yeast-related ones. And he's a really smart guy, and he has tons of awesome ideas, and he can really get the ideas done, which is, it's awesome. It's great when you find that in someone where they can have good ideas and get them done. So. And I also want to thank Sven and Kevin, who really introduced me to the whole gene drive field. I had never touched a gene drive until Sven came up to me and said, "Hey, you want to try some gene drives in yeast?" I was like, "All right, that sounds good." And then Kevin has been a champion of the whole gene drive field recently. And I want to thank everyone else in the lab. It's a really cool lab. It's big. So this is from George's birthday, and I think only half of these people are currently in the lab. Probably less. But there's more people. So I think it goes, it's like a clock. This is the new people, I think. And then these are some alums as well. I think like about half or 2/3 are current people in the lab, and then there's more new people too. So it's an amazing environment, and I get to work with the smartest people I've ever met in my life, and it's always inspiring me to be better and more creative. And everyone's so nice too. Everyone's really creative and intelligent, but everyone's really nice, and that's a rare combination, I think. It makes the lab pretty special. I want to thank a lot of friends that have helped me through my PhD. I want to thank my friend Dan, who I wouldn't have gone to grad school without his support. 9 years of support or so. I want to thank Alice and Nikolai, who are here. They're my best friend, probably. We talk about podcasts, science, everything. I want to thank John and Xavier. Xavier just left for Houston with John. They're really great listeners and super nice people. And I think Xavier's watching via the live feed, so thanks, Xavier. I want to thank Dan we have the same sense of humor, I think, and it's rare to find someone who has the same sense of humor with you. And I want to thank Rich, too, for always a good time, always having fun together. And I want to thank my cat, who always provided support. And she always has a lot of wisdom from her 23 years. She's actually Jeff Buka's cat, so I don't know if A lot of people know that, but Jeff Buka gave me his cat. She's so sweet. She's very old now, though. I want to thank my family so much for always inspiring me to be better. My grandparents worked so hard to provide support for their kids. And I want to thank all my aunts and uncles, especially for teaching me how to laugh at things. They're always, like my aunt Patrick and Cheryl have always helped me with my career in science as well, writing really good tips and stuff, as well as labs. And then Leslie and Gary always providing me with a good joke. As well as my cousin Laura, who's been a great role model for me since I was like in the 4th grade. She represents what I want to be as a scientist as well as potentially a physician in the future. And I want to thank my parents so much for first inspiring me to be a scientist and for being there for me, always emotionally, and always supporting me in anything I ever did. My dad was an electrical engineer, and he kind of taught me to have the physics background I think that I have, as well as think logically. And my mom has always been great and always been there for me emotionally. So, I want to thank you. Any questions?
There are many yeast that cause infection like complications.
yeah.
Could gene drives be used? Maybe not.
I think they could be, yeah, quite well. At least with some of the preliminary results with Candida, it seems that they work quite well. And anything that has some level of homologous recombination, I think you could use gene drives to some extent. So I think they could be really useful in that area.
Obligate sexual component.
Yeah, that's a very important one too. Yeah.
Do you need Cas9 expression in the reversal drive for it to be functional?
So, I don't know. As long as there's Cas9 expression from the previous drive, that should be enough. I've seen some experiments where Cas9 has been expressed only just transiently, and Cas9 is powerful enough that if it's there at any level, the reversion should happen. So, I would think not. I haven't tested that, but I would think not, as long as it's present from the previous drive. So.
In the Yogi experiments,
Yeah.
Did you have any idea how to control for competence, like the ability of the different strains to take up oligo?
Yeah, we did a lot. I tested a lot of different techniques for transforming yeast, and electroporation across all the strains seemed to be the best for specifically oligonucleotides. That can't be, I wouldn't say that though for larger pieces of DNA. For whatever reason, oligos and electroporation go together the best. So, but for whatever reason, larger pieces of DNA, and if there's some sort of cut at the locus, lithium acetate transformation seems to be better from my experience. So, but oligos get into the cell, I found best through electroporation. So. Cool.
Okay, thank you.
Thank you. Turn off the
What's going on? Skipping out to work? I don't know.
It's my lunch break. You can Uber here. Should we say?
Work is going to seminars anyway.
No, it isn't.
You do that. You're learning. Just tell them
Nobody.
Oh, yeah, is it okay? Okay. Okay, good. All right. Thanks.
Of course you should. Yeah, it's very nice.
Sorry.
So now you won't do it when you get your head deep like this. You do it all. At that point, you'll be just like cold-hearted.
Can we go to that one?
You need to go in.
All right, see you guys.